Welcome. Epistasis is when the expression of alleles on one locus masks or changes the expression of other alleles on a different locus. For those of you that are new to science, a locus is where genes are found on a chromosome. As you can see in this drawing, the homologous pairs have two locuses. The upper locus in green represents the black bear, which we will get into more detail in the next slide, and it has the dominant allele. And the lower locus in blue represents the white bear, which we'll also get into more detail in the next slide, which has all recessive alleles. So, for our fictitious example, we will generalize by saying that there is only one species of bear, and it can vary in color from black to brown to white, depending on which alleles are expressed. We'll be going over the single dominant epistasis pathway. In this pathway, if a letter is capitalized, it represents the dominant gene. However, if it is lowercase, it represents the recessive gene. If the first locus has at least one dominant A, it would mask the expression of the second allele by not allowing the homozygous recessive AA to go through the pathway. This makes sense as A is the dominant allele, but it can mask the expression of both alleles at the other locus, even if the other locus has a dominant allele. And by the way, what I just mentioned is a phenomenon of single dominant epistasis. But on the other hand, if the first locus is homozygous recessive, it allows enzyme 1 to move down the pathway to the second locus, which is the white bear, as you can see in the picture. If the second locus has at least one dominant B, it allows enzyme 2 to be produced, which gives the brown bear phenotype. But if the second locus is homozygous recessive, it can't go any further down the pathway, and it would help express the white bear phenotype. After going through the pathway, we are ready to go over a dihybrid cross, which means heterozygous at each locus. The key states that if the genotype has at least one dominant A, then it will be a black bear. However, if the A genotype is homozygous recessive and the B genotype is either homozygous dominant or heterozygous, then it will be a brown bear. And lastly, if both genes are homozygous recessive, then it will result in a white bear. Now, when we examine the cross, we observe that black bears are in green and would make up 12 of the possible 16 phenotypes resulting from this cross. We then see that brown bears, which are less likely due to the prominence of a dominant A, are in red and only make up 3 of the possible 16 phenotypes. Lastly, we observe that the white bears are in blue, and the least likely because they need all recessive alleles, which when we do the cross out is why they make up only one of the 16 potential phenotypes. The ratio for this dihybrid cross is 12 to 3 to 1. This next cross is between a black bear, as the first individual has a dominant A, and a brown bear, as the second individual is homozygous recessive on the first locus, but has a dominant B allele on the second locus. When we work this cross out, we can see that black bears make up 8 out of the 16 possible phenotypes, as the dominant A allele affects half of the cross. We then see that brown bears make up the other half of this cross, because when no dominant A's are present, there is always a dominant B allele, which rules out white bears. The ratio for this cross is 1 to 1 to 0, as the 8 simplify down and the white bear isn't a possibility.